So, a very good afternoon, dear delegates and participants. Myself, Satyajit Mohan, Assistant Professor on behalf of Roland Institute of Pharmaceutical Sciences, heartily welcome you all to the second scientific session of Day 4 online faculty development program on artificial intelligence in drug development, recent trends and future perspectives, that is being sponsored by AICTE-ATAL Academy, New Delhi, pleasantly organized by by Roland Institute of Pharmaceutical Sciences, Berhampur. Dear delegates, now we have our most respected resource person of this session, none other than Dr. Lakshaman Bhai Dungar Bhai Patel, sir, with us. Before inviting sir to start his eminent talk, it's my privilege and honor to introduce such a magnanimous profile of Dr. LD Patel, sir, to all of you. Sir has completed his PhD from prestigious LM College of Pharmacy, Gujarat University. Dr. LD Patel, sir, currently serving as Adjunct Professor Pharmaceutics, Parul Institute of Pharmacy, Vadodara, Gujarat. Dr. LD Patel, sir, is having more than 43 years of academic, industrial, and research experience. Sir has got more than 139 research publications of national and international repute till date to his credit. Dr. LD Patel, sir, has received Professor B.M. Mittal Memorial Award for Pharmaceutical Education on his phenomenal article published in IJPER. Dr. LD Patel, sir, also been respected by being nominated for Global Teacher Award 2020 by AKS Education, New Delhi. Sir has also been nominated for International Scientist Award 2021 by VD Good, Coimbatore, India. One of his poster was selected as best poster for oral presentation at 62nd IPC 2010 held at Manipal on December 2010. Sir has been awarded first position in pharmaceutics at Akhil Bharatiya Vidyarthi Parishad 7th Farm Vision 2016 Vadodara. Sir also has been credited guiding 178 number of MPharm theses and 14 awarded PhD theses under his guidance. Sir has filed and applied 6 Indian patents and published 4 international books. Dr. Patel, sir, has received a whopping amount of 47 lakhs as project travel grants from various authorities. Dr. Patel, sir, has received Modrov's grant of 26 lakhs as co-investigator for a computer lab development in the year 1989. Sir has completed R&D project of AICTE of grant of rupees 18 lakhs as a co-investigator and got awarded A grade excellently by AICTE. Sir also has received a Modrov's grant of rupees 7.50 lakhs as co-investigator on modernization of laboratories of pharmaceutics and farm technology by AICTE in the year 2004. Sir has also participated in 1 week 6th International Neural Coding Workshop held at Madborg, Germany, and presented a paper on his scientific work. Dr. LD Patel, sir, has been an expert to AICTE, MSME, and Pharmacy Council of India. Dr. Patel, sir, he is having research interest in the area of development of novel drug delivery system, computer and software statistical applications, direct compression formulation and technology, dissolution enhancement, sustained release dosage form, transdermal dosage forms, artificial neural works and applications, Six Sigma, QBD, biostatistics and statistical tools and technology applications. With this brief introduction, once again, I welcome Dr. LD Patel, sir, onto this virtual platform and request sir to come on board to proceed with his scientific session. Over to you, sir, please.
Okay, thank you very much. I'm a teacher and I want to remain a teacher. I'm audible, everything okay? Hello?
You are audible, sir.
Yes. Okay. Is PPT?
Just yes, it is full screen, sir.
Yes, full screen first. Okay. Can I keep video on or it is? Hello? Can we keep video
off, sir? No.
For that,
Off
I have
also
to
no
go
issue, sir.
again. Just a minute, I have to go
You can
there.
off your video.
Why? Because it takes that
Yes,
stop
yes.
came.
More
Okay.
network. Yes, yes. Okay, sir. Continue, sir.
So,
Thank you.
okay. Thank you. Thank you, Principal Ravi, sir. Thank you, Dr. Viswa Mohan Sahu, Dr. Satyajit, staff members of the most reputed, highly reputed RIPS, Roland Institute of Pharmaceutical Sciences. I'm very much thankful to the AICTE and ATAL to give me a chance to interact with the participants. I was also one of the participants in the phase 2, in the previous phase. Today's talk is Introduction of Artificial Neural Network, in short it is ANN, and its application in pharmaceutical sciences or research as a part of online FDP at RIPS. These are the three outlines of my presentation. So what are ANN, how they work, and in which areas they can be used, and how we can use. So I have taken one case study with a screenshot of a software. We can have a live software also, but that takes too much time online. So I took short screenshots and presented in the slides. So we know that in pharmacy, QBD is one of the important approach. And I'm not going into the detail of that QBD. So what is QTPP, what is CQA, what are the different stages of QBD, and where DOE, design of experiment, is used, and how it is, any formulation is optimized, and then it is validated, and then what other interpretation we are doing in the life cycle of the product management. So that comes under the QBD. Now, for QBD, we have data. We have so many steps, and at these steps, we are getting data. How that data should be treated. So, again, important component for QBD statistics, they are DOE. Six Sigma is also one of the part of that, and ANN. There are so many softwares available, MATLAB, Minitab, SPSS, and others. They all have almost regression, t-test, ANOVA, ANN also, neural network also, in so many softwares. So statistic is to be interpreted, is analyzed, and then interpreted using these softwares. Going to the artificial neural network. Now we know that intelligence is God gift, and it is not same to all. It may vary from person to person. So my intelligence is at different level than the others. And again, it may vary from time to time. My intelligence at the age of 35 and at the age of 69, it's a different. So intelligence, a God gift, vary according to the time also. And how human learns. We as a human learns by experiences, How computer or how any software can learn, any machine can learn. So I'm not going into the different type of the intelligence in AI and so on. So I'm going directly with what is machine. And we know that there are more than 100 billions of neurons which are related with the intelligence. And not only 100 millions neurons, they are interconnected. One neuron is connected with so many. An average connection is more than 10,000. One neuron is connected with other 10,000 neurons. So its brain is a very complex and no one can directly replicate it. But scientists, uh, computer people, they are trying to replicate some part of that we say as a simulate or a mimic. What is AI? So that was regarding the intelligence. Now artificial intelligence. It is the ability of any machine. Artificial means machine. It's not a human. It's not alive. Or a computer. For what? To think and to learn. First learn, then think. Then again learn and think and have the use. So learning and its application is in AI, intelligence. To be an intelligent machine, it must have some different characteristics than the human. It may try to mimic the things, but there should be something different. Why? Because live thing is a different thing and machine is a different thing. So that we know will not go in detail of that, but biologically, what is neuron that we see, that we study at our level. So first is in ANN, first subtopic is AI. Then what is biological neuron, how it works, what are artificial neuron, which in terminology ANN we say as a node. Then everything is dependent on some functions. Functions are to be imbibed in the cell of that nodes. So that functions are known as activation functions. Then how computer and brain differs, how ANN and computer differs, how computer ANN and brain differs. So that things. And lastly, in introduction part, what is the difference between statistical methods and ANN methods? So we'll summarize all the 7 points, not going into detail what is thing required for today's study that I will speak or I will highlight. There are so many things on ANN. If we want to learn it in a depth, then it may take more than 2 years. So I have to be summarized in that things. AI, before AI, what is neural network? Neural network is artificial representation of the human brain to mimic or to simulate its learning process of the human brain. So this is a representation, not exactly copy, one thing. Second, ANN, they are interconnected group of artificial neurons, and that is nodes. There are so many artificial neurons like brain neurons that are used for computational purposes as a model with some functions imbibed into that cells, and according to that functions we are getting our output. Third important thing with AI is that ANN is an adaptive system. Adaptive means which has tendency to change its structure based on the information or based on the exposure. Like human brain, if a kid is exposed with the table, 10 times, then he may remember that things. So that is a learning. And for that learning, neurons may be changed, not biologically, but with some content, with weights or so many things. And so that nature of the biological neuron is known as adaptive system, and that is utilized in artificial neural network also. As information fed to the computer or to the node or to the artificial node, it may change, or there in biological neurons, if some input is given, it may resist that input up to some threshold limit, and after that it changes. Similarly, some threshold limits can be given in ANN. ANN learns like human brain. ANN learns by examples. By example, some data are given. That data it study. They are the data are exposed to that network with the functions, and weight are adjusted accordingly. It learns, and after learning, it gives the response also. And And suppose in Excel, we are using that predictive value of y. What is predictive value of y using regression analysis? Similarly, also here, ANN gives the response after learning. It gives the response. Artificial neural network, they are made up of artificial neuron. In short, they are known as nodes. As a basic processing elements. Each node is a processing element. Each node works as a processor of a computer. Each node works as a processor of a computer. You understand that? And in different node, we may imbibe or we may program different functions also. So depending on the requirement, that can be done. How biological neuron works? It receives some stimuli. It receives some input up to some threshold. It doesn't respond, but after something it gives some stimulus. Stimulus may be positive or negative, excited or inhibitory also. Similarly, artificial neural, they receive a signal as an input, then they processes that input signal depending on the ANN configuration functions or number of nodes or so many things, and then converts that into results or interpretation, and we say as output. So three main things: input, processing, and output. So that is the basis of ANN. ANN need to go training. Training is a learning. In books they write trainings. In softwares they also use learning and training. And in our normal life, in human brain, we use learning. Learning is a training. Training is done for specific number of times, echoes, how many iterations, in which data set of input and output are used to modify the connection weights or strengths. Input can be a raw data or output of the first neuron, output of the previous neuron. So one neuron processes it, gives the response, gives the output. That output can be fed to the second. Again, it processes that output, feed it to the third, and so on. So, depending on the type of the requirement, ANN configuration can be changed. So, this is in basic of the artificial neurons. It mimics the human brain, but it doesn't copy directly. It is not exactly the copy of that. It mimics only certain processing capability of the human brain. ANN doesn't have any biological counterpart in the brain. They are not attached with the brain neurons. They resemble it. Their processing, their learning process or some process, they are similar to the human brain, mimics, exactly not resemble. NN terminology is derived or borrowed from neural science or neuroscience. And ANN is defined as computer algorithm that attempts to mimic or simulate the parallel, highly interactive, distributed processing similar of the brain tissue. It is a parallel process. Understand that it's a parallel process. What is serial and parallel, that we'll see in comparison part. So it's a parallel processing machine. What is a biological neuron? We study in anatomy, physiology, in pharmacology that these four parts: dendrite, soma, axon, and synapse. What is dendrite? It's an input. It accepts the inputs. What soma? It processes it. It's a nucleus or it's a neuron cell body. It processes it, and processed inputs are transmitted by axon and then given to the other node, other neuron by synapses. So this is a figure of that single neuron. Now if we study artificial neuron, the base principle is taken, and the last figure is this figure is actual brain diagrams. So synapse and so many things. There are so many interconnected things, very complex things. And same is represented in books also. So what are dendrites, what are cell, what is nucleus, and then what are the synapses. I'm repeating that there are more than 100 billion neurons in a human brain, and they are interconnected up to 10,000 with each other. So brain is a very complex. ANN can be made complex, but a computer has some limitations. Now, always the direction of the flow or data is from input to the output, not in the reverse way. From output to the input, it cannot be. Cannot be. But in human brain, we can go in a reverse engineering also. In artificial neurons, that is not possible. So that is one main difference between that things. Now we in pharmacology study that the cell membrane has minus 70 MV resting potentials. Why that is a resting potentials there? Why? Because cell contains fluids and it contains so many ions, potassium, sodiums, and we know that proton pump inhibitors, PPI, and so many things. So they maintain the electrical resistance of the cell minus 70. If there is some input, if there is some stimuli that then that resting potential is disturbed. If stimuli is positive, then resting potential increases, and if it is negative, then it decreases. But when after some time it gives some action, there is some change in the action potentials, in the resting potentials of the cell, and we get a pulse, we get a response. So this is biological neuron. Similarly, in artificial neurons, some threshold is given. Some threshold is given. So basic principle is same. Again, I'm not repeating. So this is the first figure of the neuron, and it was given to the world by MP McQuillos Pitts. He simulated the brain functioning with some artificial neuron and given these things. Here again, there are some two symbols. One symbol of sigma. So this is sigma, and this is as activation functions. Sigma is known as a summation functions. Let's start with this. So this is our x1, this is our input, this is input to x2, this is x3, and this is xn. Then due to neuron potentials, there may be some weightage of that. So here we write W1. So weightage of that x1 is something. Similarly, W2, W3 and Wn. If we have multiplication of W1 into input 1 plus W2 into input 2 plus W3 into X3 and so on, we'll get some value, and that value is known as a summation function value, and function is a Wixi. So this is a simple node. Then we may change a functions here and we may get a required out. Now, so function part are more important. Functions parts are more important. And what are the functions used in the softwares that we'll see, and what actually biological tissues work, how they work, what will be the output functions of that, that we'll see also. Each cell, each neuron in artificial ANN neural network work as a single processor, and all neurons, they are interconnected. They have adaptive interaction between the elements. Adaptive means they may change their weights as per the input. Now these are different functions. Let's take y is equal to x, y is equal to 2x, y is equal to 3x. So that's a linear functions. In mathematics, we say that's y is equal to fx, or y is equal to 2x plus 3. So it's a linear functions. We say as identity functions. In software, they say as identity functions. Then second is linear threshold. It is linear, but after something. Suppose here x increases, y remains 0. This is x and this is y. When x reaches to c, again x is 0. After c, as x increases, after acquiring c value as increases, y also gets increases, and that function is y is equal to fx. So this is a linear functions, and this is also a linear function, but it's a constant. So that is known as a linear threshold. So some threshold limit is given here. Similarly, we may have binary steps threshold, two types. Up to this, then again it's constant, then again it's something, then again goes, and so on. So binary, ternary, and so many things. Then function may be of bipolar, one pole, then constant, and then second pole. Similarly, piecewise linear. Some part is linear here, then it is linear with second relationship, it is linear with the third relationship. So it is a piecewise linear. So there are so many functions. I'm not going into detail of that. We are related with 10 H exon functions and sigmoid functions. Sigmoid function is e raised to x divided by 1 upon 1 plus e raised to x. If we simplify that, if we divide all the terms, numerator and denominator, by e raised to x, so e raised to x by e raised to x, it comes 1. 1 by e raised to x, that becomes e raised to minus x, and e raised to x by e raised to x becomes 1. So sigmoid functions, exon functions, is widely used in our cell working, and hence it is important. And we say it is a logarithmic functions. It's a e means we say in log ln ln 2, and e value is 2.731 something. So this is biological part. Similarly, we may have hyperbolic sigmoid up to sometimes. So this is a simple curve of that, and I drawn it using Excel. Suppose x is 0. Here if we x 0, then 1 divided by 1 plus e raised to 0. E raised to 0 is 1. So 1 plus 1, 2. So 1 by 2. So it comes to this. Similarly, if x is 1, minus 1 and E is 2 point something, then we calculate, we get fx this much, and I plotted it and got what books have given. So this is that sigmoid. Similarly, here also we may have that according to the data. So my idea is that there are functions, and functions are the mathematical expressions, and they are very simple. They are the right one. If we have a different or mixture of functions, then that activity may be a different. So these are the different functions. I'm not going into detail. These are the functions of the MATLAB software, which is widely used in ANN. MATLAB has so many modules of ANN, and this is our that sigmoid functions. This function is a sigmoid function. The return is now in books log is also written and hyper is also written. Hyperbola, that is a logarithmic part. So log sigmoid or hyper sigmoid or x sigmoid, that's same. That's same. So here they written log SIG. Log SIG. That functions is widely used. This tangent functions is also used in pharmacological softwares. So this 10 functions is also widely used. So these two functions, exponential functions, are used. Let's take one simple example. So this is a artificial neuron representation, figurative. In computer, in that cell, in the circuit, in that circuit set, it may be somewhat different. But this is a theoretical written in a books. I have taken the sigmoid functions. Okay, so W1, W2, W3 that I took, X1, X2, X3, they are our variables. In variables, then we get summation functions, W1 into X1 plus W2 into X1 and so on. And then depending on we get the output. Let's take one. Now this is also important thing. The average firing frequency of biological neuron follows a sigmoidal characteristics. This is principle written in so many pharmacological books. How firing frequency of neuron works, that gives a representation of the sigmoid functions. And it is non-linear, it's not a linear one. So sigmoid functions have some biological basis and it is widely applied in calculations. Let's take example. Let x1 is, we have three inputs. X1 is 1, x2 is 0.5, x3 is 0.3. So these are our x1, x2 and x3, our inputs. And let's a single summation function means wi sigma wi into xi, simple functions. Okay, and that I written here also. So 1 into minus 0.2, 0.5 into 0.4, 0.3 into 0.25, 2.5, and that gives this value. So our y will be this if there is only summation functions. Suppose we have then added a linear functions. Linear functions means y is equal to fx, y is equal to x. Linear functions means y is equal to x. So here at the end we are getting 7.705. If y is equal to fx, then output will be 0.705 if it is a linear function. But if it is a sigmoidal functions, then I calculated it. Okay, this we calculated. Then this I calculated here is a blue color, and that gives 0.6693. So there is a combination of summation functions Okay, this is a summation functions, and this, sorry, this is a summation functions, and this is a output after sigmoid functions. So here in one neuron may have this part, in second may have this part. We can have, according to the complex nature of the process, we can go with increasing number of layers, number of nodes, and can mimic the brain partially. So that scientists are doing the things. Brain and computer, they are not same in so many parameters. One brain is 1/10 the calculator. In calculations, one brain is equal to 1,000 supercomputers. Now computer can do one activity one time. It can get multiplication or whatever is that. But brain, I'm teaching you, and my brain is with my grandson, is writing here, that also. So brain neurons can work in a complex functions, while artificial neuron or computer cannot. In memory, computer can win. Why? Because now there are terabytes, gigabyte, and so many things. But in brain, in human brain, as exposure is more, as it is more utilized, as ages, as it get age, it is weakened. Memory gets weakened. But in brain, in computer, it's not so. It remains same. Sometimes it may hang if we are using so many functions at a time. So in real world, brain is superior in memory. In real world, arbitrary computer is superior. Arbitrary. In storage, it's okay, but in real things, brain is superior. Brain must get experience through learning. Through learning, it must have some learning. If I know 16 into 4 is 64, then and then I can write that. But computer, we have to program it. We have to give that functions in one of the node, and then we can get it. While in brain, we get by experience. Teacher, in nursery, they teaches the boys and write A, capital A. So that is a experience. Same not is with the computer. Computer has a program. So there are so many things in computer books and pharmacological books and ANN books. I summarized one important is working part, and that is more required. Computer works in a serial, one task at a time. Now our brain, human brain, it works parallel. It's a parallel processing. We can think so many things at a time. So it's a real world or perceptual world. ENN, they also have parallel, but 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 one term at a time. There are so many things, but all not at a time. Summation can be done, then exponential can be done. So there may be parallel processes there, but working, handling of task is once only. Why? Because it is a computer. But in brain, we can have so many things at a time. I'm writing, I'm looking into the note of my grandson, and I'm teaching to you also. So that's a human brain, and that's a God gift. Okay. So there are so many things. I'm not going into detail of that. Why? Because this is the introduction part, and I think we covered more than 30 minutes in that. Yes. So this is difference between stats methods and ANN. both share the concept same. Their mechanism is somewhat same, like probability and logistic, hyper, hypo, so on. That can be possible in statistics now also. Difference is in term of weight determination. ANN have weight change. It's adaptive, so it can change the weight. How, that we'll see in one example written in my hand. There are no assumption in in ANN. In statistics, we have so many assumptions. It has independency between the variables. ANN has independency between variable. In statistics, it's not so. ANN can learn by self, statistics cannot. So these are the main differences in statistical methods and ANN. But ANN are based on statistical functions or mixture of statistical functions. Now, there are some difference in abbreviation or some terminology in statistics as well as in artificial neural network. In statistics, we write model, we write equations. What is the model? Quadratic model, second order, linear or order. In ANN, they write network. So network are depending on that modeling. Estimation, that is word in statistics. We estimate it, while here it is learning and then predicting. Regression, that is a statistical word. In ANN, we use supervised learning. Interpretation, interpolation, generalization, and so on. We are not related with that in depth, but at least model and network are required to understand. So the introductory part, what are ANN that we seen, how they work, basically input, process, and output, how neural network differ from the computer and brain, how they are similar to brain, like parallel processing, and how they resemble brain in two aspects. One is learning, and second, connection strengths of the neuron, connectivity between the neurons. So that we study what is ANN that we did. Now we did only introduction part, not in depth. Now how they work. Working is very complex and there are more than 1,000 books on that, and it's very difficult to summarize it, but I tried my best to mimic that things in short. What is the structure of ANN? How they are designed? What are the layers? Why? Because in designing without layer, that complex nature is not possible. And why there are intra and inter layers? In layers or in between layer. Inter layer means within the layer nodes. Okay? Inter, in between. And intra means inside that. Learning how ANN learns. And what are the laws? What are the learning principles or that configurations? And what are the models or what are the network functions? This is a general figure written in so many books, not only one or two. It is written in more than 200 books. The color may be different. In all, we have three layers. One is input layer. One is input layer. Okay. Here I written only three. That book has written only three. It can be up to n. Then hidden layers. And then output layers. This is a second hidden layer. If required, there may be one hidden layer also. Now see the connectivity. That's a magic. That is the essence of the ANN. So this is our first node. first variable, it is connected with each one of the hidden node. uh, and this strength is W1, then so many things. Second is also interconnected with that. Then there may be some functions here. It will give output, and that output again feed to the second hidden layer. Here we are using some functions and that gives some calculations. Here other function is used, it gives something that. So to make it very complex, this can be done. So that is the logic behind the ANN. So I'm not going into detail of that things. So these are the three things important: structure of the network, and for that we need number of layers. How many layers we have? Two layers definitely there are, input and output. We may have hidden layer, 1, 2 or whatever's, from 0 to what we need. Then property of nodes. Property of nodes means functionality variability of the node. How that x1 node may change, -1 to 1, 10 to 15. So range of each node, it may be different, like variable. Suppose I'm taking sodium lauryl sulfate as a variable, and I'm keeping the range from 0.5 to 1. So range is 0.5 to 1. But if we have lactose, then it may be different. So range of each node may be different. Then activation functions or transfer functions. Again, that's a property of node. Each node may act as a processor of a computer. Then design. This is also an important thing. Why? Because learning methodology depends on that. And again, it also involves some functions that known as activation functions. I'm leaving this part. Yes. So basically, three things: input, hidden, and output. Okay, what is that? I'm not going into detail things now. Yes. Okay, that is here. This is important. Number of neurons in hidden layers, they are depending on the trial and error method. It may have 5, it may have 2, or it may have 10 also, depending on the requirement of the functions and complexity of the configurations. To each neuron, if we feed too much, then that is known as a overfit. Suppose we give some homework to a child when he comes from the home, from the school at home. He may tolerate, but suppose we give over homework, then what will be the reaction? It may have some reaction. So overfitting may deviate the normal functionality, may generate the problem. And ANN will have that problem of generalizations. So if we feed too much, then there may be some difference in the required output. So that is a limitation also. Okay, useful to this. Now, interconnection and intraconnections. They are connected with each other, and they are also connected with within the layer nodes. Again, connection may be fully, connection may be partial. We'll see in figure somewhere when there it will be. It may be on one side only, and it may return also. So it's a backward. So unidirectional, bidirectional, fully and partially. I'm not going into that part. Yes, now learning part. This is important. Brain learns by experience. Similarly, ANN machine learns by some data, some algorithm. Something is given to them, and from that it learns, and their weights are adjusted, and then it will give desired response. So weight value are important in strength connections. Learning of the data is the essence of the neurocomputing. So that is innovative as compared to the by themselves. And reinforcement learning, something is different from the above two. Generally, supervised and unsupervised are used, but reinforce is nowadays used in learning. What is a supervised? A teacher is present during the learning process and it gives the exact output. My y will be this, my result will be this. Your marks should be greater than 35 to pass. To get this 60%, to get first class 60% and so on. So it provide a training data set and a result and a result. And that set is known as the input and and the expected output which is given, that is known as the correct output. So that is in supervised learning. In unsupervised learning, teacher is there, okay, he is there, but he will not help. He will not give output. He will see that you are getting output, okay, good. He will not give the value of output, okay, with some conditions, with some conditions. And reinforcement learning, here here input is given but no output. Output is given either at the end that you are correct or not, you are correct or not, you are right or wrong, you are achieving or not. So that is a reinforce, and generally in most of the softwares it is used. What is online offline that we are doing since so many years, these are the learning rules, how that works, which functions will be there. So HEP rules, Hopfill rules, delta rules, difference rules, that is known as the least mean square error, and SOFF rule. So I'm not going into detail. Generally, we are taking a LMS, least mean square methods, in computers. Xi minus x bar square, sum of, then divided by n, and so on. Now, these are the models. Depending on the equation parts, we have so many models. And blue color, they are widely used in MATLAB, Minitab, even in SPSS also. Multilayer perceptron. What is perceptron? We are going into see. So at least you must see this, read this. But what is Illman network? No need to study. It is available there. We will see somewhere in one of the slides. What is MLP? You remember the name. What is that? No need to go in detail. Why? Because we are not the configuration organizers. We are only applicable part. We pharmacy peoples. So this is a single layer perceptron. Input layer and output layer. How they are connected? They are connected fully. So it is in a unidirection, one direction flow. Then what is a perceptron? Perceptron is a neural network term. It is a model term in ANN given by Rosenbelt, right? Rosenbelt in 1957, that he named a mathematical formula that receives inputs and give computations and give output. That formula part, that is known as a perceptron. So this is a single perceptron. huh? Formula is summation function, WIXI or whatever that's. Sum of that give the answer. So a single layer neuron with a simple functions. And that was devised by Rosenblatt. How it works that we studied X1 into W1 and that we did. Then multi-layer. There may be hidden layer, one or more. And from input layer to the hidden layer, there is connection strength, and that strength is given by some matrix. So this is W1, this is W2, and this is W3. Similarly, from this is first node hidden layer, this is second node, or that there are again three W21, W22, and W23, and so on. And from that again, if there is a connection with output, then again there will be made weights there. And these weights, they are adaptive depending on the threshold, depending on the given condition in the equation. Suppose y is equal to 3 up to x is 3. Then if x is 4, then it is 3 point something, and so on. So as per the requirement, that function can be designed, and that may be involved in the hidden layers. So input layer, hidden layers, and output layer, that part is known as a MLP, multi-layer perceptrons, and we are going to use that here. What is in detail, that I'm not going into that things. Okay, yes. Now this is a recurrent network. Here, before that, let's see that. Without that, you will not understand. So this is only single layer. X1, X2, X3, and our Y1, Y2, Y3 input layer, output layer. Here we have hidden layer like the previous figures, and we are getting that. So it's a multi-layer things. Now, if this Y1 is connected back with some condition to X1, or this partially or fully, or one of this Y, it is feeded back to something, then it is known as a recurrent things. Recurrent things. Again, recurrent may be fully or partially. Here it is fully. Feedback links. It is returned and then again so on. Repeated till we get the answer. We get the correct answer, or nearer answer. Okay, so that is the things. Now at the end, all functions, they give their optimization or output depending on the R value, depending on the mean square error, MSE, and mean absolute error. So in all, they are calculated at the end. Let's take simple example how it will work. In example we studied, x1 is 1, x2 is -1, and x3 is 2. And weights, W1 is 0.5, W2 is -0.1, W3 is -0.2. Similarly, for second neuron, it is 0.6, 0.1, and 0.7. Okay? Calculate that summation functions. We calculated as a summation functions. This into this, then this into this. So that we did. I'm just repeating it here. Okay, and that comes to 0.2. So this is a summation functions. Again, so this value by summation functions. Now that act as an input for this node, and where the function is again different, and that function is the exponential functions. So if f is 0.2 in exponent, we get answer 0.9. Sorry, 0.55. Okay, 0.55. So this 0.55, it's exponential functions. So this result is achieved by exponential functions at the hidden layer neuron. Okay, similarly here also we are getting 0.71. Summation is 0.9 and exponential for second node is 0.71. See, there we taken only one example. Here I've taken two. Similarly in computer it may be more than 10 also. So that is summation function and exponential function. Again that hidden layer is connected with the output layer with some strengths or weights. And again that function is also exponential functions. Then we will get answer 0.70, 0.478. Okay. If we do by hand, then it makes too much time. But computer will do within a second. Now this is our predicted y by computer. Suppose actual y is 0.4, then we will get, if actual y is 0.4, then we get the value. This is 0.478, this is 0.4. So we'll get value difference as an absolute difference. If here we give some threshold that if it is less than or less than 0.6, again that is feedback. That response is feedback back with all. Again repeat it, get till some actual y is obtained. So that is the complex mechanism in artificial neurons. So that is these things. What are the deciding factor? That weights, number of hidden layers, number of nodes, and data set of the training. I repeated this. Yes, I did by my hand, by our calculator. Same things in that thing. You see this? This is very complex things. It may take more than a day also, but you can understand that we are getting the results. So this is not first echo. This is a second echo. I calculated further by using some principles. You don't need to remember it, but just see it. Have your things. Okay, how complex it is. So this is second iteration. This is third iteration. Go until we get the desired results at the end. So that's the complex part of the ANN calculations, and it's not easy to configure it all at a time. So ANN is a good statistical tool. I think it is 1.5. So within 15 or 30 minutes we will complete the parts. So these are the application parts. It is widely used everywhere. It can be used, but we have to identify the functions. We have to identify the number of layers. Why? Because if we provide too many layers, then it may become a complex things. It is used everywhere in many fields, including engineering, psychology, medical chemistry, diagnostic, pharmaceutical research. It is used for classification. How patents are classified, how signature is verified, how medical diagnosis is identified nowadays by robot. That AN is used. Is there any risk by that person in giving loan? Is the same voice is mine? So voice recognition, image recognition we use in a mobile also. So this is that pattern, that methodology that uses application is known as a classify. It classify the given inputs and match with the correct one. If it is matching with some deviation, some justified deviation or some given limits, then it says yes, otherwise that mobile will not open. Then modeling. So in robot that is used in process control and so many things, molecular modeling, even in docking also ANN is used in our chemistry. In predictions, I want to predict my Y, this is used. In data associations, correlationship and regressions, optimization, self-organizations, And this each small words, that is one article. That is one article in literatures, in journals. In automotive industry, warranty activity analyzers. So it's a one article of 11 pages. So these are the used of ANN in different segments of life, in aerospace, in automotive, in banking, in defense, in electronics, in engineering, in finance, in insurance, in manufacturing, in medical, in pharmaceutical sales, robots, etc., and so on. You will uh, get tired if you collect all the data and classify them. It's very complex. But so many people said did this and I copied partially. So this is little history of AN and how it is evolved and who were that uh, gold brain that brains. What are the advantages? It is universal function approximations. It gives the exact value, approximate value. It has adaptivity. It can change depending on the given threshold. Wi can be changed according to the given threshold limits. It can work with linear and non-linear and complex. It may work with so many variables. So many variables. If there are large number of variables or parameters, ANN will work, but our Excel or something, it may take so many times in processing. Fault tolerance, that is one of the important things. If one of the neuron is damaged or blocked or their connection is changed or damaged, then whole system will not collapse like computer. That part will remain dead, but other part will give the result. So there will be slight deviation, but not total stoppage. So that is a good thing. Why? Because each cell is a processor. If that doesn't work, it will not affect others. So fault tolerance, that is the greatest advantage of the artificial neural networks. These are the again addition. I'm not going some limitations. It cannot explain. Explanation is given by teacher only. So we will be required everywhere. Yeah? So that is limitation of ANN. GIGO. We study in economics also in that. Okay, what we give, what we take according to our thinking will be. So there will be GOGI. Overfitting is a problem. If we provide too much pressure to the kids, then definitely there may be some change. So overfitting. Similarly, here if data are not proper and they are given so many data, then their functionality may not be reliable. Work better when data set is sufficiently large. Sufficiently large words is more important. Not too much. Require an extensive amount of training. Without training, we can't get the testing. So training is given to the nodes and weight are adjusted by them, and then we give our test data set. Then and then we get our results. Without training, ANN will not give the response or results. Now, how we can apply in one of the model? We are going into one example of 15 minutes. Just suppose we want to use ANN. I'm giving pharmaceutical example. Similarly, there are pharmacology also, cognostical also, chemistry also, docking also, so many. I'm starting with this, and this is an introductory lecture, so I'm not going into detail of others. So first is data preparations. We must organize our data as per the requirement. Select input and output. What x1, x2 is are required. That selection is on us. Then network modeling we have to select. And which network, multilayer or that, and we have to select the learning methodology, supervised, unsupervised, or what. In so many software, generally reinforcement is default. So check the default method and then change accordingly or select accordingly. Then we have to select the number of hidden layers, how many layers we need. In pharmaceutical calculation, what we did at my PhD students level or PG students level, in my thesis is we use not more than 2 hidden layers. Why? Because results are not at so accuracy if we use more layers. So 1 or 2 layers, maximum 2. How many neurons? That depends on the X1, X2, X3. It depends on the type of X1, X2, X3. Then learning network. Then testing batches results in interpretation that we have to do. That will be C. So for that, I have data set of 3 raised to 2 factorial design where x1 is SLS and x2 is 280. -1, 0 and 1 are the level of the x1. Similarly for x2, and I prepared a granule using both, and I measured the angle of repose of that. So I got the result of S1 batch, that is 32, S2 batch, and so on. Then C1 and C2, these are the 2 checkpoint batches, that is batch number 11 and 12. Up to 9, it is a factorial design, 3 raised to 2, so 9 batches. So first 9 batches, they are the factorial design batches, and last 2, they are the checkpoint batches. I want to validate what we achieved, what they learned. That will be validated using C1 and C2. So we will feed data of S1 to S9 to the algorithm, that ANN software. Weight will be adjusted as per the threshold, and then we give the testing C1, C2, will get the output of AR. So this is our practical output. We'll get output from the computer. Okay, so this is a data set. Here in pharmacognosy, X1 may be solvent, X2 may be some other things. Similarly in docking also. So forget that X1 is SLS and X2 is 280. Consider your variable and keep your mind here to understand it. Then, yes, then download Neuro Solution. Neuro Solution 7.1 version. It is a pre-version and demo version. It takes only 5 minutes to download. So download this, install in your computer. It is an Excel friendly software. So this is an Excel sheet screenshot. And here in your Excel, this will not be till you install this Neuro Solutions. So without Neuro Solution, this will not be there. We have in Excel, we have home, insert, layout, formula, data, review, view, and then help and so many things, or Acrobat, what it says. If we install that Neuro Solution, we'll get this part. Okay, so click on that. And now before that, let's understand the important things. What are the important things? We have to take the data. So these are our data. We have to take that. These are these first 9. They are only for training. They are for training. These are our experimental results. They are for training. And checkpoint basis, they are for testing. Checkpoint basis, they are for testing. Okay, so keep these points in mind. Download it and open your Excel sheet. Check is Neuro Solution words there. Okay. Then C, we have tag data. Then we have train network, and then we have test network. So these four things are required. Okay. So this is a data set, and this is a neuro solution friendly, Excel friendly neuro solutions. Then click on tag data. Click on here. Click on that. Click on that. We'll get 17 things. There are so many things. What are that? As input, as training set, as testing set, add this, add this, add this. So click on that and see this. That is not readable. I think I again gone on Photoshop and cut paste and got these things. Okay, so on tagging, after tagging, what we have to do, we have to select, again I'm repeating the things. So download Neuro Solution 7.1, prepare your Excel sheet with your data. It may be 2.2, it may be whatever steps. No need of factorial design. You provide your data and provide your checkpoint or what you need to check or what is your output. So then select column x1, x2, x1 square, x2 square, and x1, x2 as a input. Select this. Go with the on Excel file with your cursor, and we are selecting in this way. So select that. Sorry. This first column, second column, you keep your cursor and just click. It will this is a photo, actually not Excel sheet, otherwise it may. Then when you select it, it will be turn into blue color, light blue color. Okay, so turning light blue color, confirm that columns are selected, and that columns we use as input. That is our input column. Our response is output column. Here we can take x1, x2 also, but I want to use quadratic nature of that x1 and 2 and relationship between x1 and x2 also. Okay, so we can take that as a experimental design matrix. So select that column, required column, it will be blue color. Then click on column as input. Then click here. Okay, so what will happen? It will get dark blue color. See how they design the software. This is a light blue, means you selected it. Then again it turns into dark blue. It indicates that is tagged. Your column are tagged as input. So that completes the input part. Then what is your desired column desired? What do you want to achieve? What you want to achieve? So select that Y, keep your cursor that Y column, and then it will turn into the again blue color. Before that it was colorless. See, here it was colorless, and here it turns into blue color, means it is selected. Then click on desired, again it will turn into dark blue. Okay, so tagging of columns is done. Tagging of column is done. Okay, column as input and column as desired. So that completes the tagging part. Now select the training data set. Now training data set we took as a S1 to S9. So that are row. So select rows S1 to N9, S9, S1 to S9, they will be again turned into light blue color. Light blue color. Okay. From this we can see why this was a blue color again previously. S1, S2, S1 not. Why? Because that column was not selected. So when we select the rows, it becomes blue color. Okay. Then click on training. Why? Because that is our training data set. So click on row as training. So as such, it is very simple. But once when we first time downloaded and installed it, it took more than a week how to do these things. It was very difficult. Why? Because that detailed procedure is given, but we pharmacy people are very nauseous people. We can't read in detail. We want to do it. We want to do it, but we will not read it. So we read it and then did it. Okay. So training. Similarly, again it will change the color. Then testing. Testing is our checkpoint basis, that is C1 and C2. Select that and again click on row testing. Okay. So first we selected the column, then we selected the response desired, then we selected the rows, then we selected C1 and C2s. So that ends the selection part. Now network part. Now click on build network that we seen in the first slide. In first slide, everything is there. Okay. So click on build network. Click on build network. Sorry, yes. In there we will see that जो 14 हमने देखे थे, what models we have, MLP, GRNN, that regression MLP, what MLP you want to select. So I want to select custom MLP. Why? Because we may change some selection data. Okay. And they return it. If you go download the software and read the things side by side, you may see that in classification MLP row if greater than 1000, then select this. Otherwise, have custom network. So click on custom network. Okay. Then click continue. So network is selected. Then click continue. Then in MLP, again we have that things, multilayer perceptron. Multilayer perceptron. Okay, so click on that. Again, go with the next. After clicking, click input variables. We have here 5 variables: x1, x2, x1 square, x2 square, and x3 square. Okay, so input are 5, output is 1. There may be 2 also, angle of repose and second. How many training we are given? We are giving 9. So this is a part where we have to take precautions. And how many layers, hidden layers do you want to keep? Keep 1, get the data, keep 2, get the data, which are best accurate, select that layer. That can be done. Try and error. And then click on the symbol double arrow. Okay. So that is a selection of the input part. Then hidden layer. Then functions. Okay. And then learning part. So you will not, it will be boring and laborious to you just now. So I'm going as such and getting the results. Then have the supervised learning selection part, then configuration part, and click. After clicking on that, click build will get the network build it. Then save it as a SID. It will be saved. Now we selected test and train, but we haven't got it. Then Then here we'll click on train network. We have data, we selected, it is already selected, just we have to give the functions. So train the network and click OK. Network will be trained, weight will be changed and optimum weights will be achieved. Okay, click OK with flow. Then click test network. Uh, when we click test network, this echo graph will be, we can see it. It will be drawn by computer. So these are the number of trials where this MAC is, here MAC is this, in second trial this much, this second, third this much. So we given that it should be minimum. So what is the minimum MAC and when it will be? So after 14 it is minimum. So that is known as a echo or iterations. And what is that? That will computer will write. Okay, so test it, will get this curve, then click OK, then close total things, will get this output. Will get this output, and you see that this is not readable, so I taken here also. Yes, here R is 1. Our in Excel we are getting 0.999, 9792 in calibration curve, but this software is 0.99 in 5 figures, approximately 1. This is the actual AR, practical AR, and this is calculated AR by software, by AR. This is I did by Excel, and we got this AR for C1 batch and this AR for C2 batch. See the exactness. This is 3.33.07 and this is this much. This is this much. So where, which is best, that we have to interpret. And see the other data, RMSE, NMRSE, MAE, whatever that's, I'm not going into detail of that. It is 330. So this is the actual output of the ANN. So we can use where we need to predict our Y, our docking part, our pharmacognostical results. So we can utilize this. Similarly, this is also one of the good software by Australian University. It is user friendly for students. If you give your roll number and if your institute is recognized by them, and if you write that, suppose I write my Paro University, it is recognized. So it will give, I can download this software freely. If it is not, then you can download it, but it will not work in your in your computer. This is the link. You go in Google and write University of Adelaide Neural Network. You will get that link also. I'm not going into detail. This is the screenshot, how to download it, what are that, how to work with this, that I have given here in detail, like that ANN. I'm not going into detail of that. And this is the output. So this is the summary of the 3 outputs by Neuro Solution 1, by University Adelaide software, and by Excel. Where R value is nearest to 1, that we have to select. Okay, so this is the use of that. And there are so many articles on ANN, and it's a very growing field. Those students of 12th, they must go on this artificial neural network area. Why? Because that is the future area. Everywhere autonomous automatic industry is there, robots are there, machines are also automated. So ANN use will be tremendous. Smartness is required, and these are my words for these reputed institutes, Roland institutes. Uh, there is a good teamwork among the staffs and principals and management, and they got a good success in lesser time. Similarly, here also in our industry also we can do. This is my disclaimer. I'm responsible what I said, I taught, but I'm not responsible what you understand and what you learn. My intention is not to criticize anyone at anywhere, and this is the general disclaimer that I use softwares, images, tables, some data from university uh, examination papers, and so many things. So uh, I'm very much thankful to those contributors from whom we use that, and our purpose is, my purpose is only educationals. This is my guide at PG level, at UG level, and at PhD level, and at academic level also. But now he is no more. He expired before a year, in 1.5 years, in the initial phase of Corona. Dr. M.C. Goel, he was principal at LM College of Pharmacy. I'm very much thankful to him. Why? Because when I joined in 77, I was a student. My brain was blank, but he did so many things to me. These are my appellations. I'm very much thankful to all participants to be with me at RIPS. Thank you all. If you have any questions, you may have after de-sharing the slides. Uh, and I'm very much thankful to the Roland Institutes and Management, AICTE and ATAL platform to provide me this opportunity to interact with you people. My last principle, that is LDP, learn, digest and practice or perform, and be happy in your life. Yes, sir. Thank you, sir. Hello.
Thank you, sir, once again for your nice presentation. So this is the real things you have explained very clearly. So from the basic level to application level, so we can apply for our artificial intelligence. It will be very useful for the participants, sir. So
Yes, sir. Yes, sir. Yes,
yes,
sir. So I given the initial background of ANN,
yes,
how
yes,
it works, basic things, and then how it can be applied. My intention is that our student must use this,
yes,
even
yes,
staff members. And again, my request, my email address I have given. If anyone has problem in his thesis or her thesis at any level, PG or US, PhD level, and want to use ANN, they may contact me by email, and then
sir.
we can contact by WhatsApp also. But I'm not
Yes,
giving my
yes,
WhatsApp number, sir. Why? Because it is misused, and they
yes,
ring
yes,
at mid time. Uh,
yes, yes, yes.
so
There is
many
not good.
students,
Yes, sir.
not desirable. But those who deeply want to learn, they to contact with me by mail. Then we will give them. Okay, sir.
Yes, yes, sir.
Thank you once again, sir.
Sir, we have received some questions and comments on chat box.
Yes, sir. Yeah, I'm ready to give the answers. Chat box. Yeah, just I will see it also. Yes, so I'm starting with the Mamta. Mamta, okay, explain. Thank you. Atal, that is these things. Okay.
Some are asking, is there any software for calculation? So already you have
Yes, it
explained
is.
these things.
Yes, sir, I given the link also. Huh? You go on Google and write neural network software. You will get so many. Okay, and for students, this Australian university software is very good. Okay, so that is one question. Second, is there any related with the any doubt part in
Yes,
the
sir.
understanding?
Somebody
Yes.
is writing, ANN is unable to use in pharmaceutical research.
No, sir. I used in my PhD thesis. We publish articles in elsewhere also. They are online articles. They are open articles. It is available. You can see also. So it can be used in our research. Even so many industries they are using in research. Not only in research, in manufacturing also. Now ANN is used in robotics manufacturing. Uh, so Sundar sir, it is used widely, but but our mindset should be ready to adapt it. That is required.
Yes.
Why? Because our industry don't want to change their methodology part. If they change their method or instrument, they have to change the SOP. They have to train the workers. They have to write to the authorities and so many things. So they are not ready to use that. It can be used. It can be applied, sir. Yes. So, yes, Sundarsan, is it okay?
Yes,
They were,
yes, yes.
yeah.
Yes, he replied, sir.
Okay. Then, is there anyone?
I think not there, sir.
Okay. Thank you all for your nice feedback. But my request is that you can contact me by mail for your problem, not only in Excel, even in mathematics, in biostats also, in Excel software also. Okay, using t-test and so many things. Okay, sir.
Yes, sir. Thank you, sir. Satyajit,
Thank
sir, you
you.
can proceed.
Sir, sir. Thank you so much, sir, for sharing your immense knowledge on artificial neural networks, comparison between computer and brain, structure, design, then put focus on reinforcement learning and on ANN models. Thank you, sir, for recognizing us the artificial neural network systems linked with the pharmaceutical subjects. The entire session was lively, engaging, educative, and worth-watching because of your benign presence. May I now request our respected principal, come convener of this e-faculty development program, Professor Dr. B.B.B. Ravikumar sir, to present our eminent resource person, Dr. LD Patel sir, with an e-certificate of appreciation as an expression of our gratitude and respect from entire RIPS fraternity.
Yes.
Sir, thank you very much for accepting our invitation and sharing valuable knowledge about artificial neural network in pharmaceutical research. And as I told you at the beginning of this session, you are really inspiring. Last FTP was also, your feedback was very nice. Thank you very much, sir. Keep guiding us whenever we need some um, help.
Yes, sir.
We'll contact you, sir.
Yes,
Please help us.
sir. You can call me at any time for any type of work for your institutes, for staff and students.
Thank you, sir.
Thank
I
you,
will
sir.
be with you. I will be with you.
Sir, thank you.
Thank you.
And on behalf of the entire fraternity of Roland Institute of Pharmaceutical Sciences, I would like to present this e-certificate of appreciation. Kindly accept, sir.
Thank you. Thank you. I'm accepting heartily and very much thankful to our students uh, and to principal sir also.
Thank you, sir.
Thank you, sir.
Thank you.
Thank you.
Thank you, sir, once again.
Okay, so I'm leaving, sir.
Yes, sir, yes, sir. Thank you once again, sir.
Okay, thank you. You can contact me for any type of work, educational
Sir.
work. Don't worry. This one,
Yes, sir,
or
yes.
for any student also.
Thank
Yes,
you,
sir,
sir.
yes, sir. Thank
If
you.
they
once
have some problem in understanding or interpretation of such types, then you can. Okay. Okay. Thank
Yes,
you
yes.
and good luck.
Thank you.
Stay safe and
Thank you.
secure.
Thank you, sir.
Thank you once again, sir.
Dear delegates and participants, with this, we officially conclude the second scientific session of day 2 of day 4 of EVT, so sponsored by ATAL Academy. The next scientific session will resume today on the same platform very soon. Thank you all, dear participants, for your kind attention and participations.